The overall goal of this procedure is to describe the development of a multicellular 3D organotypic model of the human intestinal mucosa cultured under microgravity provided by rotating wall vessel bioreactors.
This method has a wide range potential as a tool for discovery in both health and disease, including interactions with pathogens, antigen trafficking, and inflammatory processes. The main advantages of this technique are the mimicry of the phenotypic diversity of the gut and the use of rotating well vessel bioreactors that allow efficient diffusion of nutrients and oxygen.
Start by unscrewing the cap from the fill port of the culture vessel and adding 50 milliliters of rPMI. Once the vessel is full, replace the cap and wipe any spilled medium with 70% ethanol. Allow the vessel to incubate for at least 15 minutes to overnight at room temperature. When ready to continue, use the fill port to discard the culture medium and add 30 milliliters of the 3D culture medium to the vessel. Replace the cap on the fill port and again wipe up any spilled medium with 70% ethanol. Remove near-confluent flasks containing human umbilical vein endothelial cells and human colonic fibroblasts from the incubator and wash them twice with PBS. Then, detach the cells by covering them using a 0.25% trypsin solution with